again for all joining. It's my pleasure to introduce Michael Lane and Lana. I'm going to butcher her last name, Zholudeva. How is that, Lana?
Actually.
Oh, thumbs up. Woo. Anyways, it's so cool to have them because I met them in Cleveland, Ohio, as they reminded me, in 2019, and they were doing some presentation on some research that they were doing with higher level spinal cord injuries. And I don't know, it kind of inspired me, for lack of a better word, because you don't see a lot of research that is coming out for higher level, especially in respiratory function specifically. So finally, after years, I've been able to connect again with them, and they're here today to talk about the research that they're currently working on. So thank you, Lana and Michael, and I'll hand it over to you.
Right, thank you very much, Terry, and thank you again for the opportunity to be here and talk about some of the work we're doing. I'm just going to start off talking a little bit about the background behind some of the work my team's been doing, and which leads into the ongoing collaboration that Lana and I have. So I'm going to talk to you a little bit about my research, but feel free to interrupt at any time. Just interject and you can start talking, or you can type in the chat, and I'm happy to take questions throughout the presentation to make it more like a discussion. So the focus of my research, as Terry mentioned, is on cervical level spinal cord injury. Being a preclinical scientist, we study this in animal models. So we study what happens after these experimental cervical injuries to better understand what these injuries do to the spinal cord itself, what potential there is for what we refer to as plasticity, which is the ability essentially of the spinal cord to make new connections or essentially result in a little bit of functional recovery, and how we can go about designing therapies to enhance that. The focus of this research is primarily on breathing. As you all know, injuries at high levels, at cervical levels, compromise a huge range of functions, but among those is breathing. Which is obviously life-threatening, but it's also it leads to often assisted ventilation, a need for assisted ventilation, and it changes quality of life very dramatically. So we're looking at trying to better understand how breathing is affected, and how we can develop therapies to promote better breathing, recovery of breathing, and essentially repair of the underlying networks, the networks of cells that control the muscles that allow us to breathe. And we hope that in doing so, we can then start to carry these treatments over into other systems and start being able to treat not just respiration, but many other functions. So I'm first going to talk a little bit about the way we breathe. Obviously, we know we all breathe for a specific purpose, and that's getting air in and out of our lungs, oxygen into the body and carbon dioxide out. And essentially, one of the beautiful things about this is it's a really simple behavior. We often don't have to think about it. It's autonomic, it's automatic. And so these muscles just contract away and we breathe. And as you can see here in this diagram, that big green thing underneath the lungs is the diaphragm. And that's probably the most important muscle within respiratory networks. And that's the one we study in our research. The diaphragm is essentially controlled by what we call the phrenic network. And the phrenic motor system makes for a great system to study because we can learn a lot about it, as I'll show you. The other thing to understand is that we can control our breathing. You can choose to do this if you'd like to, you don't have to, but if you look at this diagram and you breathe in as the image inflates and you breathe out as it deflates, then straight away you can see that you've completely changed the way you've been breathing. So you've gone from not thinking about your breathing at all to having complete control over your breathing. And the really cool thing that this illustrates is we are now using different networks, different series of cells in the brain and spinal cord to control our breathing. And this speaks to somehow the way that we can tap into different parts of the network or different neurons that can facilitate this plasticity. So the fact that there are multiple different ways to control our muscles means these are different systems we can harness with treatments when we look at trying to improve breathing. So this diagram here looks a little bit complicated, but what I essentially want to show you is that in this image of a person, you have the muscles essentially that can contribute to breathing. So there are lots of different muscles that can contribute to breathing. Chest muscles, the muscles between the ribs, and of course the most important, as I said, the diaphragm seen under here, which sits underneath the lungs. Interestingly, when we look at these numbers here, this is showing you the distribution of neurons and nerves that innervate these muscles from the spinal cord, from cervical levels to thoracic levels, all the way down to even lumbar levels. So all throughout the spinal cord, there are neurons that innervate respiratory muscles. So injury at nearly any level can affect these muscles and change the way we breathe. With that in mind, if we have a mid-thoracic injury, then the muscles between our ribs, the intercostals, may not work as well, and the abdominal muscles may not work as well. And these are actually important muscles for expiration or respiratory behaviors like coughing and sneezing that can actually be partially compromised with even a thoracic injury. With a higher injury at cervical levels, we start compromising even more of these networks. And the most important one is compromised now too, is the diaphragm. And that's why under most circumstances with cervical level injuries, people often require assisted ventilation. So they may require a ventilator. So as I said, we study this preclinically. So we use animal models to study this. And what I'm showing you here are just diagrams of a human cervical spinal cord and an animal cervical spinal cord. And what we can do is start to model the types of injuries that occur in people, as you can see here on the left, and we model these in animal models, which is what my research focus is on. And essentially what we try and do is develop models that have similar types of anatomical deficits to what we might see in the human population. This is showing you an image of a human spinal cord after an injury at cervical levels. And the degree of compromise in the spinal cord here sort of recapitulates what we can achieve in our animal models. So it's important that we relate this back to our animal models, so we're studying something that's relevant to the population of people living with spinal cord injury. The other really nice thing about the phrenic motor system, which controls our diaphragm, is that it actually provides us with a system that we can identify all the components of. And that's really important if you want to start understanding how to repair something, is to be able to tease it apart and understand what all those components are. So within the phrenic system, there are neurons in the cervical spinal cord called phrenic motor neurons, which innervate the muscle, the diaphragm, directly. And we can trace these and we know where they're distributed. In fact, this is a section of a rodent spinal cord here. And this is the phrenic motor neurons we can actually label with a tracer, so we can map where they are. And that means we can see how they change their connections after an injury as well. We can also look at descending axons that come down from the brain and the brainstem. And these are centers that control our breathing. And when we can stick a dye also in these cells and see where the axons go to. So in this particular image here, we now have our phrenic motor neurons, which control the muscle, in green, and they're receiving input from axons in red. And so just this one image shows you the network that enables your diaphragm to contract with every breath. The beauty of being able to use these techniques is we can now see how that changes after a spinal cord injury. What's compromised, how does it change? And that will tell us what plasticity is. What little events might happen that can allow us to use therapies to harness these changes and improve breathing. In addition to this, we can start mapping out other populations of neurons. And the reason I raise this is because these cells are distributed all throughout the spinal cord. They're called spinal interneurons. And these are cells that essentially can relay information down from the brain and can also control these motor neurons that innervate the muscle. So these interneurons are a very different population of cells that can make new connections very easily, which means they're quite plastic. And we can map the distribution of these in the normal spinal cord and see how they change after an injury. We can see how axons coming back in from the periphery might innervate this network that we've now identified. And we can do this again by using dyes to trace these afferent axons coming back in from the periphery. And we can look at populations of axons from a range of different cells in the brainstem of the brain. So by using these different dyes in different colors, we can essentially color code all the different components of the network that allows us to breathe. So that gives us the anatomy of the network that controls our diaphragm. And we can study this, as I say, very effectively, which is one of the strengths of our ongoing research. We can then start to study the function as well. And so looking at breathing function, there's two things we need to measure. That is, how do we breathe? And then we can do that by simply measuring the air we breathe in and out the amount of air we breathe in and out, and how quickly we breathe. And these are called basic physiological parameters, or parameters of ventilation, when we measure how we breathe. But another measure of how we breathe is how the respiratory muscles contract during each breath. And that's where we start to understand how the diaphragm itself might be compromised by injury, whether it's not contracting properly with each breath, and how we can find strategies to improve diaphragm contraction to make the muscles work more effectively. And we can record directly from the muscle in order to measure that. So what happens to this network after spinal cord injury? So what we're looking at here is the same network, this phrenic motor network, after a spinal cord injury, and it's been color-coded. So in green are the neurons of the spinal cord, which control the diaphragm. We have descending input from the brainstem in blue, and in red, you're seeing where this injury is. It compromises this network. So that means we lose cells that normally contribute to diaphragm contractions, and we're compromising those descending axons, those fibers coming down from the brain and brainstem. When this happens, and we record from the muscle, we can see that on the side of the injury, the muscle doesn't work as well. So it doesn't contract the way it's supposed to, which means the lungs don't inflate properly. And that's why many people need to essentially go onto a ventilator for ventilatory assistance. Now, there is instances where maybe they can recover a bit of ventilation or breathing, and they might not have to stay on the ventilator. And that's great. That's actually a sign of plasticity. And so by studying these changes in function and mapping this plasticity, we can then start to see how the underlying network, those components, contribute to that plasticity. Which brings me back to spinal interneurons. These cells are very plastic and make new connections quite easily. We can start to see how they feed into these new networks that allow us to breathe differently. Now, a lot of research that we've done and others have done have shown that spinal interneurons make new connections very easily. And we can see how they change over time, as this diagram shows you here. These green axons are entirely new connections that occur after an injury. They might not have been there before the injury, but they can make new connections after a spinal cord injury. And we can see how those changes occur using those dyes. This image here is showing you some of these components. So we're looking at the neurons within the spinal cord, and we've color-coded them. So in red are all the different sorts of neurons in the spinal cord in the middle. In pink, you're actually looking at just the motor neurons, those cells that innervate the muscles. So all these other cells here are spinal interneurons. So you can see just in this one tiny section, there's thousands of different spinal interneurons, which means there's lots of different types, but we can tap into these therapeutically. So for the interest of our discussion today, my research focuses on several of these. We're just going to talk about one type of cell. But essentially, we believe spinal interneurons are key for this plasticity and key components of the network that allow us to now use therapies to enhance function. If we can harness these cells and guide the way they make new connections, we can make plasticity even more effective, and that's what our therapies are trying to do. The question, of course, given that there's thousands of them, which interneurons do we look at? So as I said, we're just going to focus on one. And this is actually showing you a spinal cord that's been cleared. So we were able to image it in 3 dimensions. And these red dots in the middle of the spinal cord here are lots of cells called V2A cells. V2A is a cell you may have seen a little bit in the popular press. It's been in some news articles have been written about these cells because they've been shown to contribute to plasticity after spinal cord injury. So we're interested in studying how they change their connections and how treatments can be used to make them connect better. So our research team, in collaboration with Lana's research team at Gladstone, is essentially trying to understand how we can therapeutically target these cells. And we do so in 2 ways: rehabilitation, so physical therapy and activity-based strategies, and strategies for repairing the damage to the spinal cord. I'm just going to talk for a couple of seconds on rehabilitation and the ways we go about doing that. So in order to treat plasticity and provide activity, increased activity to an otherwise damaged spinal cord, there's lots of ways you can go about doing that. And we divide it into 2 main areas. The first is neural interfacing, which means we can stimulate the networks that exist in the spinal cord. And we can do that with epidural stimulation. And these are probably things you've heard of. Transmagnetic stimulation. FES here is functional electrical stimulation directly to the muscles. Or they can even put electrodes straight into the spinal cord for intraspinal stimulation. All of these essentially represent neural interfacing or electrical stimulation of these underlying neural pathways. You can also do this with physical therapy, activity-based therapies. There's a lot of work being done looking at treadmill training, which can be weight supported. There's research being done at several institutions around Canada and the US and Europe that's looking at how locomotor training can improve function. And then the respiratory system, there's also a little bit of clinical research being done to see how we can promote breathing. And this is actually really cool because all you need to do is essentially change the different gases that people are breathing. Or it's almost like lowering the level of oxygen in the gas we breathe in, like going up a mountain. So there's less oxygen in the air, we actually start breathing more, and those muscles start working harder. So from a physical therapy standpoint, we can use strategies like this to start driving activity in these pathways more and strengthening not just the muscles, but also all the neurons that innervate those muscles. We can change their connections using these strategies. And we've actually shown that by taking this, we can start increasing connectivity from the brain onto our spinal networks, and we can increase the connections that these spinal interneurons we've identified make. So those V2A neurons, we can start targeting those using these sorts of therapies, enhancing their connectivity.
Before we move on, I just wanted to jump in real quick and say, in particular with the epidural stimulation, the recent work that I think maybe has hit the press, I'm not quite sure, but it's actually hinting at the fact that when you do epidural stimulation, at least this is in animals, you're actually tapping into these V2A interneurons, or at least one part of the circuit that you're exciting more than other circuits or more than other neurons are these V2A interneurons. So it's really interesting how lots of different labs are all converging onto this cell type. And Michael's lab, I think, was one of the first, I think back in 2016 or 2017, where it was just this random study that, very randomly, serendipitously, but this cell type appeared in, the work that we were doing. And then we sort of started to see other labs starting to talk about V2As, and it's coming back full circle with these epidural strategies that are now going into clinical trial. If you've heard of the Onward trial, for example, that they're reporting that they're also tapping into the class that we have been really fascinated with, the V2As. So it's really interesting how everything just kind of converging from different sources. So I thought I would mention that.
Yeah, that's a really good point, Lana. Thanks for jumping in. But of course, in addition to that, in addition to looking at these strategies for increasing activity, there is other deficits that persist. And so essentially that leads us into this other avenue of research, which is mechanisms for promoting spinal repair. And this is where I'll hand it over to Lana to continue.
Yes, thanks, Michael. Yeah, and the reason why we would even start to think or consider repairing the injured spinal cord, the anatomy itself, is because the rehabilitation strategies that Michael just went through, either locomotor just rehab or stimulation, those strategies, they plateau at some point. Because they really just tap into the circuit, the substrate that's left, that's preserved after injury, but the underlying cause of the deficit of paralysis remains untreated. So while you're trying to work whatever you have left harder, or maybe even optimize the function, you're still not really treating the underlying cause of the deficit. You're not going down and really trying to repair the circuit. And so that's why I think both rehabilitation and repair should go hand in hand. And we're very privileged in being able to study this on a very basic cellular biology, preclinical models, where we can, have a model of injury, repair it, and then, look at the tissue or do electrophysiology or do all of that, and look at that end product and aspect, and really ask ourselves, is this strategy working? And if it's not, why isn't it working? And we can do it over and over and over again. So I think that we're very privileged in that case. So with that, I wanted to talk to you about some of the repair strategies. But to do that, we have to consider what is the cellular biology, what are the cells and the molecules that comprise a spinal cord. And so this schematic gives you an idea that the spinal cord, has these neurons, the main electrical communicators, has some astrocytes or glial cells, the supporting cells, and it has extracellular matrix, or the proteins that hold the tissue together and that serve as cellular, communication. And with a spinal cord injury, where this is the normal architecture of the spinal cord, especially with something that's like a traumatic spinal cord injury, you have this blunt force trauma that damages the tissue, damages the blood vessels. The blood vessels themselves, they release red blood cells and lots of molecules that are not supposed to be in the central nervous system. And of course, that results in a huge inflammatory response, the cell death of the neurons that don't regenerate. But over time, you also see this almost like a wound healing response that the spinal cord does. So whereas, you still have these inflammatory cells come in and trying to clean up the mess that the spinal cord injury itself resulted, you also see this, cellular response where specific glial cells, for example, they upregulate their activity and essentially form what's called an astroglial scar, or the scar, the spinal cord injury scar. But we know that this is an important part of the wound healing process, because preclinical studies in the lab showed that if you are to just get rid of this quote unquote scar, it actually results in a much more severe spinal cord injury. So in essence, this wound healing is incredibly important to keep these immune cells at bay and not completely wreak havoc on the nervous system. So we also know that as time goes on, you have this chronic state, right? So chronic state of inflammation, where the wound healing response isn't really dampening down, despite maybe the inflammatory response maybe decreasing a little bit. But this is what we believe as scientists. We believe that this is the result or the cause for the neurons for being incapable of regenerating and growing back to the original targets and communicating the way that they used to. And maybe you've heard of strategies that focus on eliminating or at least dampening down this glial scar, or dampening down the little small molecules. Maybe you've heard of CSPGs. And there's a couple of studies actually, or maybe even a clinical trial, I think, that's just being launched on targeting these CSPGs. So I think the most important thing to keep in mind is the fact that this underlying biology, right, that's the thing that drives from preclinical research to clinical research to even some of these clinical trials that you might have heard of. And so understanding that underlying biology is incredibly important. And understanding the underlying biology is also important to answer some of the questions that we consider when we think about repair and repairing with cell transplantations, for example. So one of the important questions is, who are the cells to be used in order to actually repair the spinal cord after injury? And we can think of it as both neural cells, the neurons, glia that are normally residing in the spinal cord, or the non-neural components. So I mentioned, blood vessel rupture. So one way to repair the spinal cord is to attack the vascular cells or repair the vascular cells. Another way is to, as I mentioned, look at the extracellular matrix and either dampening down the extracellular matrix with drugs or increasing it in other cases. And even with that, going down even further, there's different types of neurons and different types of glia that we can consider in either making in a dish and then transplanting, or essentially attacking them within the spinal cord injury network itself. And so when we use cells or cell transplants to repair the spinal cord, as scientists, we try and conceptualize the questions and the ideas that we have to repair. So two concepts that I wanted to briefly introduce is either thinking about spinal cord repair as a bridge concept, which is essentially just transplanting either cells or molecules that make the environment growth promoting and growth permissive, or it's a concept of a relay. And this concept is more or less thinking about transplanting cells that are inherently rich in neuronal populations. So for example, inherently rich in spinal interneurons, which we know to play a role in plasticity. In reality, it's usually a combination of both a bridge and a relay concept. And you can actually see it, whether that's from clinical trial data or from preclinical research. If you're transplanting something like an embryonic spinal cord, for example, which has been done in humans even, you're transplanting something that's growth promoting, growth permissive, but also inherently rich in those interneuronal populations. So transplant like that can do both, create a bridge as well as create a relay. And so it's an important question to ask of either the investigator or even the news media article that claims that they fix the spinal cord by transplanting stem cells. Okay, well, what was the point of the transplant itself? What kinds of cells did they transplant? Were they trying to create a bridge or were they trying to create a relay? So all of these concepts should formulate some questions that you can ask. And of course, on the stem cell note, I wanted to briefly tell you that you might see media articles that use a lot of different terminology, whether that's stem cells, precursor cells, lineage restricted progenitors. And it's so important to realize that these names cannot be interchanged. And that's because the biology tells us that they're not the same cell. So if you're looking at an article or a trial that uses stem cells, you should know that stem cells have specific properties. So these cells can self-renew, they can differentiate into all cell types. If it's called a stem cell, that means it can also turn into skin cells or bone cells, and that's not what you want in a spinal cord, right? If you see a news media article or about a preclinical study that uses precursor cells, you should realize that these have a limited potential for differentiation. So these cells can divide and they generate most cell types, but they can only be of neuronal lineage. So they can only make neurons or glia, and so on and so forth. And I think it's very important to keep in mind these definitions when you start, and inevitably you will start to hear a lot of news media articles and coverage for using induced pluripotent stem cells. And these stem cells, they are very interesting. They have an origin of not neurons at all. So they have an origin, and usually it's skin origin. And these skin cells taken from patients undergo what's called a de-differentiation process to create a stem cell. And those stem cells are then taken and made either of neuronal lineage or specific interneurons or specific glial types. So I think all of those things are really important to consider and question anytime that you hear exciting results.
Yeah, I think the field of spinal cord injury research is trying to be better about that, trying to be better at using the right terms. I think in the past, people use terms like that interchangeably, and it's been misleading. So people have not been clear about the work they're doing, all the clinical trials they're trying to establish. I think we're getting better at that now. But scientists, clinicians, we all need to be held accountable, and that's why Lana and I are very careful to try and explain the words we use and stick to appropriate definitions that won't be interchanged.
And ask the appropriate questions. So, who are the cells to be used? What is the intended target? When would be the best time to treat with the cells that you have in mind? Where should they be delivered? And why would you even consider a cell therapy a valuable treatment option? So these are the questions that with any study, with any cell type, with any product that either we take from other labs and test it in our labs or create ourselves, these are the questions that we are constantly asking every single time that we go back to the bench and start to create an experiment or do an experiment and gather data. So in terms of who the cells that we want to create in order to actually repair an injured spinal cord, we come back to the basic anatomy of the spinal cord, which you see here, with red cells being neurons, the main electrical communicators. And as Michael mentioned, there's hundreds, if not thousands, of different types of neurons that exist in the spinal cord. And I think we are just, I don't know, we're just grazing the iceberg when we start to talk about the different phenotypes, because there's just so much research being done out there, and every single new study comes out with new definitions and novel discoveries. So I think it's a very budding field for just trying to understand the types of neurons that we have. Of course, there's also the glial cells. Here in blue, you're looking at astrocytes, a very particular glial cell type. And in white, you're looking at blood vessels. And the reason why I wanted to show you the normal spinal cord and what it looks like is because we also know what it looks like after an injury. And so essentially, as you see, the neurons die and they do not come back. They don't regenerate after death. You see this glial response, right? The wound healing response that I briefly mentioned of those astrocytes trying to prevent immune cells from wreaking havoc and coming in and destroying preserved tissue. Obviously, blood vessel rupture, and essentially a hole in the spinal cord. And this is what the spinal cord injury looks like for the rest of life. Whether that's in people or in rats as well. And we take that data and we take this fact and build questions to ask of the therapy that we're considering. And essentially what we try and do is come back to this basic anatomy and try to repair it and try to come up with the particular cell types that will give us neurons, that will give us appropriate glial phenotypes, and essentially allow us for reopening the window of plasticity to maximize the function that we've lost. As I mentioned, there are other questions to consider. So what is the intended target? Are we treating bowel and bladder? Are we treating locomotor targets? Are we treating respiratory function? And as Michael mentioned, the circuit that we focus in the lab is, for many reasons, is the phrenic circuit, which is the diaphragm, and essentially the function that we study is respiration. When is the best time to treat? So that's, considering, do we go in and transplant these cells acutely after injury where this heightened immune response is happening, or do we go in and treat the chronic state, which is a whole different can of worms? Where do we deliver the cells? Do we put them straight into that cavity, or do we put them somewhere in the preserved circuits to maximize the chances of these cells to survive or to modulate inflammatory response, etc.? And lastly, why is cell therapy a valuable treatment option? We like to think that cells are smart, so it's not a drug, right? Well, I guess it's kind of a combination of everything. So a cell is a biological entity, and being a cell and being a biological living thing, it can sense the environment around it, and it can adapt, and it can decide, what to do in the environment that it's in. They've been shown to have benefits both preclinically and clinically. So cell transplantation has been shown to work. It's been shown to improve motor and sensory as well as autonomic functions. And we especially think that the scientific community in general is now asking better questions, and we're rapidly advancing our understanding of cell therapies, the types of cells we should be using to repair specific intended targets, and of course, when and where to deliver these cells. Lots of work being done. And one of those, one of the work that's being done is working with human pluripotent stem cells and creating interneurons, much like what the ones that Michael described, the spinal interneurons. And essentially, I just wanted to briefly tell you about some of the things that we do to create these cells in the dish and then to transplant them back into an animal model. So we can take stem cells, human stem cells, we can give them small molecules, essentially the food that they eat, and we can control the food that they eat in a way that makes them both of neuronal identity as well as of spinal cord identity. And we can even control the particular phenotypes of interneurons that they develop in. And one of those phenotypes are the V2A. So this is the work that I'm doing now in San Francisco, where I'm creating these V2A spinal interneurons from human pluripotent stem cells, and essentially, characterizing the cell product that we're making, making sure that we're making V2A interneurons, but also transplanting them back and seeing back into a spinal cord injury and seeing if they facilitate repair. And one of the other cool technologies that we're using is something that's called optogenetics, in order to answer these questions of, do these cells work? And what this technology allows us to do is essentially create these cells that are responsive to light. And I wanted to show you this, just one example of what that means. So here you're looking at electrical activity that I'm recording from these V2A interneurons. As you can see, anytime that you see a change in light, it's basically the neurons firing. It's electrical activity. And you can see that it's basically chitter chatter, very disorganized, just chatting amongst themselves. But if I turn on a light and turn it off, turn it back on, I'm essentially eliciting activity from all of the neurons synchronized together. So it's a really nice example of control over the cells that we're making, of the actual activity of them, telling them when to fire. And I think I have another, so this is an example of this, another way to look at it, just chitter chatter of neurons. And anytime that I shine a light, turn on the light, I turn on these cells and they fire all together. So it's a really nice tool to essentially, ask questions like, do these cells even connect to, the networks that we're trying to treat, or do these cells repair it functionally?
Another really cool thing, actually, on that previous image, Lana, if you look at the stimulation at the end of that recording, when Lana shines a light on those cells, you get that really patterned, rhythmic bursting of all the cells. But the beauty in that is, not only do we have control over the cells now, but you can control when they fire like that, much like we saw the diaphragm contracting. And so if you can get those contractions in that sort of rhythm, like Lana's about to show you again, then we can start to have cells that are doing what they're supposed to do in the right network. We can make them behave and help the muscle contract at the right time even.
That's exactly it. And it's great that you pointed out the respiration, because it's so rhythmic. I wanted to show you what an experiment that we might do look like. So as I said, we look at respiratory function, and we look at one muscle, the diaphragm, because it is the primary muscle of inspiration. And working with animals and doing preclinical research allows us to record activity at a very minute, detailed way. So here we actually implant electrodes into the muscle itself, and we can listen in of how the muscle contracts. And I literally mean listen in, because right now I'll show a recording of the experiment, and you'll actually hear the muscle contract with the recording being played here. So those bursts, those are actually the actual contractions of the diaphragm. And then when I switched it, I can't even hear, actually, Michael, I can't even hear this part of muscle. But essentially what I did was, first I started the recording and allowed you to listen into this part of the muscle, which works pretty nicely, pretty well. and then you barely can hear the dysfunctioning part of the muscle. And the reason why I like to show it is because working in a preclinical setting like this with animals, you can see the function in real time, and you can then re-expose the spinal cord and the transplant epicenter and control the cells that we've now put in, and while recording the actual function. I see that there's a hand up.
This is fascinating. I'm just with what you're demonstrating here. This is just in the lab, and are you using the light stimulation to trigger those contractions?
So not the ones that are up top here, but right here I'm showing you laser pointer. Right here I'm showing you what it looks like after stimulation. So this is the actual light that I turned on in order to turn on the donor cells, and while recording the diaphragm activity. So here is the diaphragm activity prior to stimulation, and after stimulation you can see it increase. So this is absolutely one of the most direct ways to essentially answer the question of, do these cells even work for the diaphragm? And here's a good example of that they do work.
Yeah,
So,
I guess there's been a question in the field of cell transplants for many years. If we put these cells in, do they connect? Do they actually function the way they're supposed to? This is showing us they do. Firstly, the cells do what they're supposed to do. They connect to a network we know, which controls the diaphragm. And even more importantly, like Lana's data here is like groundbreaking, because the muscle is working twice as well once you stimulate those donor cells. So we have cells that we can control. We can even start to entrain these so that they can perform certain functions with certain networks. And we can tailor therapies to treat different networks in this way. So it's really awesome stuff.
That's great. Thank you. I see I just jumped in with my question just when you were about to
Perfect timing.
Perfect timing. Yeah, we're super excited about these data. Personally, I'm applying for grants to really make sure that I'm not lying to myself, that I can reproduce this, replicate this, do a really rigorous study, and essentially, hopefully create a cell therapeutic product that then can go into what's called a pre-IND study, where you have hundreds, if not thousands of, rodents that will receive these cells to make sure that the cell therapy is pure, that the cell therapy doesn't result in anything that's not controllable. So we were extremely excited by these results. So thanks for the question.
So just briefly, I wanted to thank some of the people that work with me. I'm not a single person. We are a large team, and this is some of the people that work with me, both at Drexel. I'm in the Marion Murray Spinal Cord Research Center. It's our Twitter handle here. These are many investigators I also collaborate with around the US and overseas to do this work. And two key people I work with are Shelley Sakayama-Elbert, who's a cellular engineer, and Lana, who's a longtime collaborator, hopefully for much longer as well, Lana. And we are essentially leading this work on identifying these different interneurons and making cells for transplantation. This is my research team at Drexel, right after we were allowed to finally get back into one spot at the same time outdoors, post-COVID restrictions, and we're now getting back in the lab to do more work. I also wanted to thank greatly Jake Chalfin, who's an individual living with cervical level spinal cord injury locally, who serves as a consultant for both Lana and I on our ongoing research. He meets with us on a regular basis, at least once a month. We have a meeting with everyone where he joins and talks. Firstly, he talks about his own experiences, also listens into our ongoing ideas, and questions us, make sure we are doing relevant things, asking relevant questions, and thinking from a translational mindset. It's great to have him on board. And Lana?
And my acknowledgements go to both the institute that I'm in now. So this is the Gladstone Institute's building. We are 6 floors, and I work on the 4th floor here in the Cardiovascular and Stem Cell Institute. and of course, the labs that I've been through at Gladstone. So I started with Todd McDevitt's lab, and now I've transitioned to the Srivastava lab. So Deepak's been incredibly supportive and very encouraging for me, being at Gladstone, for me to really think about what I'm trying to create cell therapies, and going through and not just understanding the advantages and the advances of stem cells and stem cell technology, but also really consider the limitations. And so his mentorship, he has a lot of experience, obviously. His mentorship is key to really look and to really not lie to myself in terms of just having a positive result, but really dig deep and dig down to make sure that I'm creating something that can help. And of course, Michael at the Marion Murray Spinal Cord Injury Center, longtime collaborator. So thank you for that. And with that, I think that's the end of the presentation. So please, if you have, I know we have a little bit more time, but if you have any further questions, our emails, the Twitter information is there.